Morning everyone, myself Ms. Komal, working as an assistant professor at ISFCP College of Pharmacy. Today I am here to deliver a lecture session for the B.Pharm 6th semester students, and the subject is biotechnology. The topic of the presentation, the today lecture are brief explanation to the biotechnology and the enzyme immobilization. Now, firstly we have to know that what is biotechnology. Biotechnology is the use of living organisms or their components like enzymes or DNA to develop the products and processes, technologies for the improvement of the human health, for the agriculture, for the industry. It is developed by the Carl Erecki in 1990. He combined the genetic theory, combines biological science with the technology for the human welfare. Next is the type of the biotechnology. First type is transgenic biotechnology. Second is reproductive cloning. Third is reprogramming of the cell. Fourth is forensic biotechnology. Now we will explain that in these all the types what would be. The transgenic biotechnology include the transgenic animals. For example, the transgenic species for the betterment of the agriculture. Then is the reproductive cloning, in which we use the clone data, in which we form the colonies by using the restriction enzymes or the rDNA technology. Then is the reprogramming of the next is the reprogramming of the cell, in which we use these stem cells for the betterment of the human health. Then is the forensic biotechnology, in which we use the two techniques that are electrophoresis and the restriction enzymes. In the electrophoresis, we separate the charged particles, whereas in the restriction enzymes, we used to cut the enzymes into different parts, or we can also say it as the clones or colonies. Next are the applications of the biotechnology. The first application is the agriculture, second is for performing the various assays, third is in the medical and healthcare, then in the different technologies such as rDNA technology, DNA fingerprinting, then in the industry. After that, in the chemical and environmental applications, last is the in performing the various assays such as ELISA and RIA. I will explain all the applications one by one. First is the agriculture. For the agriculture, we use it for the betterment of the crops and for improving the animal health. We use the fertilizers, pesticides, insecticides, etc. Then is for performing the various assays such as ELISA, RIA. ELISA is the enzyme linked immunosorbent assay, and RIA is the radioimmunosorbent assay, in which we use the radio waves. Is in the various type of the technologies such as rDNA technology, DNA fingerprinting. Next is in the hybridoma technology, in the various vaccines such as rubella, hepatitis B, poliovirus, etc. Then is in the industry, we use the biofuel and biofertilizer, in which the various examples are involved such as bioethanol and biogas. And the scope of the biotechnology. The scope involves, first is in the pharmaceutical research and vaccine development, then is in the agriculture, after that in the healthcare, which is the major point, then in the industrial biotechnology, after that in the bioinformatics and artificial intelligence, then at last is the environmental biotechnology. Now I will explain it one by one. First is in the pharmaceutical research. For example, if a researcher is using a probiotic in his or her research, for example, Lactobacillus acidophilus or bifidobacteria. Then next is in the healthcare. In the healthcare, in the use of various mRNA vaccines and advanced diagnostics for the diagnostic purpose or for the diagnostic a disease or a condition. Then is in the agriculture for the pests, insects, for the herbicides, for the animal husbandry, for the animal healthcare betterment. Then is in the industrial biotechnology, we use enzymes, biofuels, biofertilizers, sustainable conditions, sustainable management. Then is in the environmental biotechnology, we use the waste management, bioremediation, etc. kind of things. Then at last is the bioinformatics and the artificial intelligence, in which we analyze the various genetic material by the various applications using the artificial technology or artificial intelligence. I will explain the topic, that is enzyme immobilization. Firstly, we have to know that what is enzyme immobilization actually means. It is a process of confining an enzyme molecule to a solid support. It can be any kind which a substrate is passed and converted into products. Substrate is here for the base material and for the support system. Then we have to know what is immobilized enzyme and why we are using it in this. Immobilized enzymes are the one whose movement is restricted, and that movement can be restricted onto a particular region or for a specific site. Then what are the needs for the immobilization? First is it is a cost efficient, less requirement of the money is there. Then is the enhanced stability. Stability is enhanced by providing these kinds of techniques. Then is the recycling and the repetitive use. We can use it not only for the one time. We can use it in the repetitive moves. Then is the protection from the degradation and the deactivation. Any kind of degraded material such as microorganisms can be surpassed from it. Then at last it is used as a controlled release system. Now I will discuss about the advantages and the disadvantages of the enzyme immobilization. Firstly, I will discuss about the advantages of the enzyme immobilization. First advantage is multiple or repetitive use of a single batch of enzymes, that we can use the enzymes in the infinitely types of the batches. Then is the immobilized enzymes are usually more stable. As we all know that the stability here is more. The third point is if the product is not contaminated with the enzyme. After that is the easy separation of the enzyme from the product. Then last is it reduces the effluent disposal problems. Now we will discuss about the disadvantages of the enzyme immobilization. First is it gives rise to an additional bearing on cost as it has the high cost. Then is it invariably affects the stability and the activity of the enzymes. If the stability is not proper, then the enzyme conditions are also improper, by which the microbial contamination or the microbial growth occurs there. Then the third point is structure changes. If the enzyme is changed, then the structure of the enzyme also changes depending upon the structure-activity relationship. After that is the diffusion limitations. Then last is the enzymes become unstable if the stability is less there. Next is the methods for the enzyme immobilization. It can be divided into the two subparts. First is the physical methods, then is the chemical methods. Physical methods involve the physical conditions or the physical parameters, whereas the chemical methods involve the chemical nature or the chemical synthesis of the various particles or the chemical reactions, such as covalent binding, covalent bonding, etc. kind of things. Physical methods includes adsorption, entrapment, and microencapsulation, whereas chemical methods include covalent binding and the cross-linking. Firstly, I will explain the physical method, in which firstly I cover the adsorption. Adsorption is a physical binding of enzymes on the surface of carrier matrix. In this, carrier can be divided into two parts, that is organic or inorganic. It requires weak van der Waals forces or the hydrogen bonding, and the example of the carrier here is silica and bentonite. As we can see the diagrammatic representation of the first part, in which the support is there and the enzymes are entrapped on the surface of the support system. Then is what are the advantage and the disadvantage of the adsorption. First is the simple. It is simple, then it is economical as it can be recycled or reused. After that, the disadvantage that are low surface area of the binding and the exposure to the microbial attack or microbial growth. Next is the entrapment. Enzymes are not directly attached to the support. It is simply trapped inside a polymer. The polymers examples are included such as gels or fibers, and it is classified into the two types. One is lattice type, second one is the microcapsule type. Here is the diagrammatic representation of the entrapment type, in which the lattice and the microcapsule is there. As we can see, in the microcapsule, the enzymes are entrapped inside the capsule, whereas in the lattice, the enzymes are encapsulated in the form of the lattice type or the lattice shape, where the matrix is there. Then is what are the examples of the lattice type? First example is the polyiaramide, then is the starch, after that is the polyvinyl alcohol. And the capsule type examples is colloidal. In colloidal, the nitrosulfurous colloidal solution is there, in which the ethanol is used. After that, what are the advantages and the disadvantages of the entrapment type. The disadvantages is that it may leak or some pores are leak here, and the advantage is it can be reused, recycled, and no type of the chemical reaction is involved here because it is a physical method, not a chemical method. Now I will explain the chemical methods which involves the various type of chemical modification or the chemical structures or the functional groups. First type is covalent type, in which the binding of enzymes or the water insoluble carriers is through the covalent bonds. Different types of bonds are there, and it involves the various type of functional groups takes place here. For example, sulfhydryl group, carboxylic group, amino group, or nitro group. These kind of groups takes place here. The advantages are the carrier is strong, so there is no leakage. As in the previous one, there is a leakage in the microencapsulation. So here is the benefit that in this there is no kind of leakage. And the disadvantage is it basically alters the conformational structure because it complies with the structural changes. Then the diagrammatic representation is here, in which the enzymes are trapped by the different types of the covalent bonds attaching here. Then another type is cross-linking. From the name, it itself specifies that there is some kind of linking there. Or the type of the cross chains, for example, in the configuration changes, etc. kind of things are there. In this intermolecular cross-linking of the enzyme, in the presence or the absence of the solid support was there. There is some type of the conformational changes or the cross-linking changes. It involves the three-dimensional cross-linking enzyme in which the aggregation takes place. Aggregation means split up into the different types or different forms. The advantage is it involves a very little resorption, high stability is there. The disadvantage is the cross-linking causes significant change in the active site. And here is the diagrammatic representation as the complexation occurs in the physical pharmacy. Here in the biotechnology also, with the help of support system in the cross-linking, the enzymes takes place by the cross-linking of the enzymes like the complexation process. Now last we will discuss about the applications of the enzyme immobilization. First application is the food and beverage industry, then in the biofuel production, after that is the wastewater treatment, medical and diagnostic devices, environmental remediation, pharmaceutical industry, textile and detergent industry. Now we will discuss it one by one. First is in the food and beverage industry in the form of jams or in the form of syrups, juices, etc. kind of things. Then in the textile and detergent industry for making the or generating the detergents, different types of soaps, etc. kind of things. In the environmental remediation for curing the environmental pollution kind of things. Then in the medical diagnostic devices for the diagnostic purposes to cure the diseases. After that in the wastewater treatment by using the sustainable development kind of things. Then in the biofuel production in the esterification kind of reactions. All for the topic. Thank you. Now we will discuss about the comparison between the methods on the basis of preparation, cost, binding force, enzyme leakage, applicability, and metric effects. Now one by one I will discuss about these. Firstly, on the basis of preparation, in the adsorption it is simple. In the covalent binding it is difficult. In the entrapment it is also difficult. But in the membrane confinement it is simple. On the basis of cost, adsorption is low, covalent binding is high, entrapment is moderate, and membrane confinement is high. On the basis of binding force, adsorption is variable, but covalent binding is strong, entrapment is weak. Again, membrane confinement is strong. On the basis of enzyme leakage, there is leakage in the adsorption, but in the covalent binding there is no leakage. In the entrapment is also leakage, but in the membrane confinement there is no leakage. On the basis of applicability, adsorption is wide, covalent binding is selective, entrapment is wide, membrane confinement is very wide. And last, in the metric effects, the adsorption is having metric effect, but only in the membrane confinement there is no metric effect, but in the covalent binding and entrapment there is a metric effect. Last is the applications of the enzyme immobilization. Firstly, we will take the enzyme that is glucose isomerase, in which we use the method of immobilization that is embedment in collagen matrix concentration. Its application is it provides isomerization of the glucose. Then we take the enzyme aminoacidase, in which there is again the embedment in the collagen matrix concentration. The application After that we will take the enzyme that is glucose oxidase, in which the entrapment in polyacrylamide gel method of immobilization is there, which provides the application of determination of glucose concentration. Then is the urease, which provides a layer in the platinum or oxygen electrode, which provides the application that is determination of urea and uric acid concentration. After that, last is alcohol oxidase, which again provides the same method, that is layer in platinum and oxygen electrode, which